Good morning, everyone.
Do you hear me, James?
Yes. Perfect. Although when Blue gets here, she will say that you sound like you're underwater.
Aha, I am now co-host.
You are now
There we
co-host.
go. All right, can people hear me or not?
We can hear you and we can see the PowerPoint that you have up, although we can't see much in it.
All right, awesome. So looks like it's 9:30. I'll go ahead and get started. So what I've got here are some practice problems. I assume it's big enough for you all to see. So if you see my screen and the problem that's on there, let me know.
Yeah, we can see peas, green pods. Hey, everyone. So we will get started as usual. Just so you know, we are recording this. Hopefully the recording.
All right, so I'm not hearing anybody chiming in, so I'm assuming you can see and hear what I have based upon the chat. So let's start with a pretty simple genetics problem. So in peas, green pods are dominant to yellow. So in this case, I've defined the symbols, big G for green and a little g for yellow. Notice we use one letter for one trait. So if you want to use Y's, that's fine. You'd use little y for yellow and big Y for green. So in this problem, we have a plant that is heterozygous, and that plant is crossed with another plant that is yellow. So first of all, what you should do to figure all this out is you should start with the genotypes of the parents. Remember, the genotypes are what genes the organism has, and the phenotype is what you see, green pods or yellow pods. So write down the two parents' genotypes, and then do your Punnett Square, and then see how often you would get an offspring that was yellow and an offspring that was big G, little g. So I'll give you a minute to go ahead and try that on your own, and then I'll show you how to do it if you're struggling. So I'm not seeing any answers in the answer in the chat for the question. So either that means that you're not typing it in or you're totally confused. So let me start by showing you how to do this. You've got a plant that is heterozygous. So heterozygous would mean one dominant allele and one recessive allele. So big G, little g would be heterozygous. Now that plant was crossed with one that was yellow. And since yellow was recessive, the only way to get yellow is to be homozygous recessive. So these are your parents. So then you just need to do a Punnett square. When you do a Punnett square, it doesn't matter whether you put the first one on the side or on the top. It doesn't matter. So you put the gametes from one parent on the side and the gametes from the other parent on the top. So in this case, the big G, little g parent can produce one of two types of gametes. Either they can produce a big G or a little g. And I'm not going to write it in, but we can also, well, I will write it in, but I usually don't. We can also write down the probability that they do that. So half the time that big G, little g parent will pass on a big G, the other half a little g. Now the other parent is going to donate either the first little g or the second little g, so it doesn't really matter. So all of these would be a quarter, so I won't bother writing in the rest of them, but this one is big G, little g. This one is big G, little g, two little g's for all of these. So what goes in the squares are the genotypes of the offspring, the possible genotypes. So if we look at this, part A asks for a yellow offspring. So the ones that are yellow are these ones. So that's 2 out of 4, which is a half. And then the other question asks for the probability of a big G, little g, and the probability of that is also 2 out of 4. Okay, yellow probably wasn't the best color to use, a little harder to see, but you get the point. So big G, little g would be a half, yellow would be a half, and so on. So again, all you have to do is figure out the genotypes of the parents and do your Punnett square. All right, so here's another one. We have the same trait as before, green and yellow. This time we have 2 heterozygous plants are being crossed. And again, what's the probability of yellow and what's the probability of big G, little g? So go ahead and take a minute to write out the genotypes of the parents, do your Punnett square, and tell me what you think the answers are. It looks like people are starting to get it. So if you're struggling, you can follow along here. So 2 heterozygous plants. Remember, heterozygous is 1 dominant allele and 1 recessive. So in this case, we have 2 of them that are heterozygous that are being crossed. So these are the parents. Again, you just need to do your Punnett Square, where you put the gametes from one parent on the side and the other one on the top. Now, since both parents are the same, it doesn't matter. So we have big G, little g from one parent, big G, little g from the other parent. And when we fill in the square, we get this. So in this case, yellow only happened 1 out of 4. So yellow is 1/4. Big G, little g happened 2 out of 4, which is 1/2. Okay. So simple enough. You just figure out the parents' genotypes and do your Punnett Square. All right, so now we have one that's a little bit more complicated. So we have 2 traits this time. We have tall, which is dominant to short, and we have green, which is dominant to yellow. So each parent, each plant will have 2 genes for the height and 2 genes for the color of the pods. So in this case, we have 2 plants that are heterozygous for both traits. So the first step you would need to do is figure out what are the genotypes of those 2 plants. So go ahead and figure that out and type that into the chat. What are the genotypes of the 2 parents? Shouldn't take you that long to figure out the genotypes of the parents. So type in the chat what the genotypes of the parents are. Okay, I'm only seeing a couple answers, but they're both correct. So I noticed that Sophie and Amia, is it, I believe, they wrote it differently. So Sophie put the G's first and then the T's, and Amia put the T's first and then the G's. I assume that's big G, little g, not big G, little t. That would be a mistake. And it really doesn't matter. So you can put whatever trait you want first and whatever one you want second. It is usually a good idea to be consistent with which one's first and which one's second. So since I wrote the T's in the question first, I'll put that one first. So, oops, sorry about that. I need to annotate here. All right, so the 2 parents are big T, big G, little g. And we're going to cross 2 of them because they're both heterozygous. So one way to do this is to figure out all the possible gametes from the first parent and all the possible gametes from the second parent. But if you do that, you end up with a big square. And I highly, highly recommend against that. There's a much easier way of doing this problem that's faster, it's quicker, and far more reliable in terms of not making mistakes. So remember, Mendel's 2 principles were segregation and independent assortment. Segregation says that the alleles separate. Well, if you understand meiosis, that makes sense because in anaphase 1, the homologous chromosomes separate. But Mendel's principle of independent assortment says that the inheritance of one trait does not influence the inheritance of another. In other words, they're independent. So think of it like this. If I flip a coin, I get a 50-50 chance of heads and tails. But if I flip a second coin, what I got on the first coin doesn't influence the second one. Those are totally independent events. So if I want to know what are the odds of flipping two heads, all I have to do is figure out the odds of heads on the first coin, which is a half, and heads on the second coin, which is a half, and then multiply. Because the probability of independent events occurring simultaneously is the product of their individual probabilities. So what we're going to do is ignore one of the letters. So let's ignore the G's. Pretend they aren't there. If I ignore the G's, then the cross is just big T, little t crossed with big T, little t. And that's pretty simple to do. You just did it on the previous question. So that's the square for the T's, for the height. So now let's ignore the T's, pretend they aren't there. So now I have big G, little g crossed with big G, little g. So again, if I do a Punnett Square, it would look like this. I'm just taking the gametes from each of the two parents and putting them on the tops and the sides. Okay? So now let's see if we can figure out the probability of what we were asked for. So in part A, we were looking for tall. So how often did we get tall? Well, that happened 3 out of 4 times. That's a better 3 out of 4. Okay, so there's 3 out of 4 for tall. And yellow was little g, so that's 1 out of 4. So how often would I get tall and yellow? Well, the answer is the product. You just multiply. And if you are afraid of math, you can multiply fractions. If you know how to multiply, you know how to multiply fractions. It's simple. You just multiply the numerator and multiply the denominator. So multiply the top, multiply the bottom. So 3 times 1 is 3. 4 times 4 is 16. There's your answer. It's as simple as that. So you do not need to use a big square. In fact, I'll show you a problem in just a little bit where I'll break you of that bad habit if you try to do it, because it really is a bad habit. You should be doing the small squares and multiplying. All right, so now let's look at answer B. B says we want the offspring to be pure breeding for both dominant traits. So remember, pure breeding means you're homozygous. You don't have any options in terms of what alleles you will pass on. So pure breeding for the dominant trait for the height would be big T, and that was 1 out of 4. And big G for the other one was also 1 out of 4. So the probability of both of those occurring together is 1 out of 16. So there's your answer for part B. And part C is an offspring heterozygous for both traits. So heterozygotes would be 2 out of 4. Now, I highly recommend that you reduce your fractions at the beginning. So yes, you could write 2 out of 4, but it makes the math a little bit easier if you change 2 out of 4 to 1 out of 2. Now, C also wants heterozygous for both traits, so we need heterozygous for this trait as well, and that's also 1/2. So 1/2 times 1/2 is 1 out of 4. And there's your answer to part C. So the key to doing a problem that has more than one trait is to treat them independently. Just ignore one and do the Punnett square like you normally would, and then ignore the other and multiply your fractions to get the answers for what you're looking for. Everybody following along with this? Yeah, don't be shy if you have questions. I'm sure if you've got a question, other people are thinking it too. So don't feel like it's a bad thing to ask questions. All right, so let's try another one. So again, we have the two traits, the height, big T, dominant to little t, and the color, big G, dominant to little g. So in this problem, we have a plant that is homozygous dominant for height and heterozygous for color. And that plant is crossed with a plant that is heterozygous for both traits. So first of all, I want you to write out the genotypes of the two plants and put them in the order listed in the question. So what is the genotype of the plant that is homozygous dominant for height and heterozygous for color? And then write down the genotype of the plant that is heterozygous for both traits. So I'm only seeing 3 answers so far, and I'm liking what I'm seeing. So the plant that is homozygous dominant for height is big T, big T. I'll go back to annotate here. So that first plant is big T, and it's heterozygous for color. So it's big G, little g. And the second plant is heterozygous for both. So heterozygous, remember, is one dominant, one recessive allele. So again, all you need to do is ignore the G's and do your Punnett square for the T's, and then vice versa. So in this case, I have big T crossed with big T, little t. And again, it doesn't matter if you put big T on the side and big T, little t on the top, or vice versa. So whatever you did is fine. And then you do another Punnett square for the color. So again, same thing. One goes on the top, one goes on the side. Okay. So now that we've got this set up, we can look for how often we got what we were asked for. So A is tall and yellow. So tall is 4 out of 4. All 4 of them are tall. So that's the same as 1, right? 4 over 4 is 1. And yellow was little g, so that's 1 out of 4. So tall and yellow is 1 times 1 out of 4. Okay, so there's your answer for A, 1/4. B, an offspring pure breeding for both dominant traits. Well, pure breeding for the height would be big T. We got 1 out of 4 for that. Pure breeding for the dominant color would be big G. That happened 1 out of 4. So the answer would be 1 out of 16. That's a 6. It's a better 6. Okay, and then C is heterozygous for both traits. So heterozygous for this one is 2 out of 4. And again, I recommend that you reduce your fractions at the beginning. Instead of writing 2 out of 4, write 1 out of 2. And for heterozygous for this one, it's also 2 out of 4, which again, I would recommend that you reduce your fraction to 1 out of 2. So 1/2 times 1/2, 1 times 1 is 1, 2 times 2 is 4. There's your answer. Now, if you had left it as 2 out of 4 and 2 out of 4, the answer you would have gotten would have been 2 times 2 is 4. 4 times 4 is 16. But then you need to reduce that fraction. So 4 out of 16 is 1 out of 4. Personally, I think it's easier to reduce the fractions to begin with.
Hey, Rick, maybe go back and do B again.
Ah, I did goof, didn't I?
Yeah.
Thank you for catching that mistake. So
It was
part
the
B,
kids in the
offspring pure
chat.
breeding for the tall was 2 out of 4, not 1 out of 4. So it's 2 out of 4 times 1 out of 4, which is 2 out of 16, which is 1 out of 8. Yep, I made that mistake, sorry. So for part B, the answer is 1 out of 8, not 1 out of 16. All right, so let's try a different sort of problem. So in this one here, we have red-green color blindness. And red-green color blindness is a trait that is on the X chromosome. So those sorts of traits we call sex-linked or X-linked. When we write out the genotypes for sex-linked traits, we must include the XX chromosomes for female and the XY for male. Notice that on the previous problems with the pea plants, one of those plants was the male and one was the female. At least one was donating the pollen with the sperm and the other one was donating the eggs. But I didn't tell you which one. And the reason why is it didn't matter. Could have been reversed, it could have been flipped, and you would have gotten the exact same answer for the problems. But for sex-linked traits, it does matter, and you must include the XX chromosomes for female and XY for male. Now, red-green color blindness is a trait that is recessive. So for females, there are 3 possible genotypes. Now, in this problem, it doesn't tell you what letter to use. So you could use C for colorblind, but if your handwriting's bad like mine, you'd have a hard time telling a big C from a little c. So I like to use B for blind. So the female could either have 2 dominant alleles, 1 dominant allele and 1 recessive, or she could have 2 recessives. Notice that X little b, X big B is the exact same thing as X big B, X little b. So yes, I understand the one in the middle you can write in either direction, but by convention, we also write the dominant allele first if it's heterozygous, okay? So in this case, sex-linked trait of red-green color blindness, it's recessive. So this individual here would have normal vision, and so would she because she's got the dominant allele. The little b for red-green color blindness won't show. Only this one here would be red-green colorblind. Now for males, there's only 2 possible genotypes because they only have 1 X chromosome. So either that X chromosome has a big B or it has a little b, in which case he would be normal and he would be colorblind. Okay? So with that in mind, let's do the problem. So we have a colorblind man and a woman who is heterozygous for colorblindness. So first you need to write out the genotypes. So for the man, we know he's XY, and if he's colorblind, it must be a little b. The female is heterozygous, so she must have 1 big B and 1 little b. So no different than before, you're just going to put the gametes from each parent on the top and the side. And again, it doesn't matter which one goes on the top and which one goes on the side. So I'll put the male on the side, X little b and Y are his possible gametes. The female on the top, X big B, X little b are his. So we have X big B, X little b, X little b, X little b, X big B and Y, and X little b and Y. So in this case, we see color blindness would have occurred in this daughter and in this son. The one on the top left here is a female that is not colorblind, but she does have the allele for color blindness. So we often refer to people who carry recessive alleles, but they don't show them as carriers. So she would be a carrier of red-green color blindness. But 2 out of the 4, this one and this one, would have been colorblind. So the answer to this question would be 1/2. So Y-linked traits exist, but there's really only one Y-linked trait that matters, and that is a gene called SRY. And that SRY gene stands for the sex-determining region of the Y chromosome. If you have that gene, it turns on a pathway, if you will, turns on a whole bunch of genes where the ultimate result is being male. So who has the SRY gene? Only people with Y chromosomes. So if you have the Y chromosome, you have the SRY gene, and therefore you're male. And if you don't have that SRY gene, then you're female. Now, it's not quite that simple, but for this level of course, yeah, it is that simple. Beyond that, there really aren't too many genes on the Y chromosome. There's a few genes that have to do with, production of testes and things like that, but quite frankly, most of them are insignificant, okay? By convention, we draw the X first. So males are XY, they're not YX, okay? Just like by convention, we put the dominant alleles first if you're heterozygous, okay? So, I mean, yes, I suppose you could write it the other way, but nobody does that. It's just not unconventional. All right, so here's another one. Again, we have red-green color blindness, so I would recommend that you use B for blind instead of C for color. In this case, we have a man with normal vision and a normal vision woman, but her father was colorblind. So see if you can figure out the genotypes of the man and the woman, and then proceed with the Punnett Square. All right, so let's see how you're doing. Seems like some people are starting to get the answers, so let's work on it here. So we have a man with normal vision. Well, first of all, I know he's XY because he's a man. And if he has normal vision, he must have the dominant allele. So X big B and Y. The woman has normal vision as well. So I know she has a big B, but I'm not sure about the second allele just yet, right? And by the way, using a blank or a dash to represent an allele that you're not sure of is a good thing to do. Sometimes you can go back and figure it out, and sometimes there's not enough information to figure it out. But using a dash when you're uncertain is a good thing. So in this case, we know she has a big B because she has normal vision, but we're not sure about the second one. But it then says her father was colorblind. So her father must have had XY because he's a man, and he must have had a little b if he was colorblind. So what did he donate to his daughter? Well, it sure wasn't the Y chromosome because she was a daughter. So he must have donated the X little b to her. So now we can fill that in. She must be X big B, X little b. So now you do your Punnett Square. So I'm just gonna, since I didn't leave much room, pretend these are here. And then we have X big B and Y for the male. So we have X big B, X big B, X big B and Y, X big B, X little b, and X little b and Y. So in this case, color blindness would only be 1 out of the 4. And notice it would always be the male, right? So it is much easier for males to end up being colorblind because it only takes 1 allele and that shows up, okay? I'm seeing in the chat, I think I'm lost. Which part is losing you? So the one thing I do see in your chat answer is you have X big BY, X big BY, and that's not quite right. It's just X big B and Y. So males only have 1 X and 1 Y, and only the X has an allele. And I know since you're in the chat, you can't do this properly, but the alleles are always written as superscripts on the X chromosome. So it really should be written the way I have it, but I understand when you're typing it in, that's not easy to do. So the correct answer for the male would be X superscript big B and Y, okay? And then for the female, X big B and X little b. So let me reiterate something that I hope you guys understand. If a trait is sex-linked, you must include XX for females and XY for males. If a trait is not sex-linked, you still need to cross a male and a female to get offspring, but you do not include the XX and the XY for the genotypes because those genes are not on the X or the Y chromosomes. So for autosomal traits, that's everything except for traits on the sex chromosomes, do not include XX and XY. For sex-linked traits, you must include it, okay? So it's possible to have a question where one trait is sex-linked and one is not. So you would put in the XX and the XY for the one trait, but you would not put it in for the other one, okay? All right, so remember I said you don't want to use a big Punnett square. So if you have been using big Punnett squares, good luck with this one. If you haven't, good. This question should be pretty simple for you. So let's say we have an organism that has the genotype big A, big B, big D, little d, and another one that's big A, little b, big D, big D. What's the probability that when crossed, those offspring end up big A, big B, big D, big D? I'll give you a minute. Go ahead and do that on your own. And I'm seeing a couple of different answers. So clearly there's a little confusion. So let's see. So again, we can take advantage of Mendel's principle of independent assortment. In other words, we can look at these one trait at a time. So one letter at a time. So the parent that we have is big A, little a, big B, and so on. But all I care about are the A's. That's the parent, big A, little a, big A, little a. So I can do a Punnett square for that. That was easy enough. Now I want to do a Punnett square for the B trait. For the B trait, I have big B for one parent, and I have little b for the other parent. And some of you who are getting a little bit better at genetics would recognize, geez, I don't really need a Punnett square for that one. If one parent is big B, they're always going to pass on a big B. And if the other parent's little b, they're always going to pass on a little b. So there's no choice. All of the offspring will be big B, little b. So now let's look at the D trait. We have big D from the first parent. And we have big D from the second parent. So there we go. So how often did we get big A? The first one here. That answer was 1 out of 4. There it is. How often did I get big B? 4 out of 4. Now again, I recommend that you reduce your fraction. If you left it at 4 out of 4, you'd have to reduce at the end. And then big D, we got 2 out of 4, which is the same as 1/2. So the answer is 1 times 1 times 1, and 4 times 1 times 2, or 1/8. Okay? I could give you the whole alphabet, and it really shouldn't take you that long to do. Now, if you were trying to do a big Punnett square, you're probably still drawing out your square. It's going to take you forever to do a problem like this. But if you take advantage of Mendel's principle of independent assortment, you just do separate squares for each letter. By the way, some common mistakes that I see when people start doing genetics problems is I will see a Punnett square that looks something like this. And this is totally wrong. What this is saying is that this parent on the left passes on genes for whatever the A trait is, but doesn't pass on any genes for the B trait. And the other parent passes on genes for the B trait, but not the A trait. Okay? And that doesn't make sense. If you are using the little squares, you should always have one letter per square. Do not mix the letters. Nor should you have a situation where you have the gametes that look like this. This is a gamete that's diploid. Gametes aren't diploid. All right, sperm and egg cells are haploid. They will have one of each letter. Genotypes are diploid. You have 2 of each letter. So to give you a specific example, you all have 2 genes that control tongue rolling. Whether or not you can roll your tongue depends upon which alleles you have. But you have 2 of them, right? If mom and dad both gave you the dominant allele, then your genotype is big R, big R. If one parent gave you the dominant allele and one gave you the recessive, then you're big R, little r. And if both parents gave you the recessive allele, then you're little r, little r. You also have 2 alleles for your earlobe shape. So some people have free earlobes and some people have attached earlobes. In this case, free earlobes is dominant to attached. So let's say that you're heterozygous for both. Are you going to pass on a gene for tongue rolling? Sure. Now, which one? Flip a coin, right? Probability is a half that you pass on a big R. Probability is a half you pass on a little r. But if you pass on a gene for tongue rolling, you're also going to pass on a gene for the earlobe shape, okay? So if we had a situation where we had a individual that was like this, crossed with another one like this, if you did a Punnett Square and it looked like this, you'd be saying that one parent passes on the gene for the tongue rolling, but not the earlobe, and the other parent passes on the gene for the earlobe, but not the tongue rolling. And that's wrong. You need a separate square for each letter, okay? And notice that the gametes are haploid. They only have one letter. Now, which one? Well, you got a 50-50 chance as to big R or little r in this case. Okay, does that make sense? All right, I'm going to stop sharing for a second and then open up a. All right. So here is a practice problem for how to do a pedigree. So pedigrees are basically how we figure out human genetics, right? So when Mendel wanted to know how his peas were passing on their traits, he crossed them. When Thomas Hunt Morgan wanted to know about his fruit flies, he crossed them. But if you want to know about a human trait, you can't just go making crosses at will. The only thing you can do is to look at what crosses have been made. So we have certain symbols that we use. We use Roman numerals to represent the generations, regular numbers to represent the individuals, and then circles represent females, squares represent males. Anybody who has that trait that's being discussed, and you can make up any trait you want, we get a filled in circle or square. So let's say 3-5 is a friend of yours. Okay, tell me about 3-5. Male or female, have the trait or not? So here's the third generation. We got 1, 2, 3, 4, 5. So 3-5 is a female. She does not have the trait. But maybe this is some nasty trait that she doesn't want being passed on in her family. So maybe she's considering having kids and wants to know, should she risk it or not, right? Clearly her brother's got this trait. She doesn't want to end up with her kids having it. So the first thing you need to do is figure out, how is this trait inherited? And there's basically 4 possibilities. It can either be autosomal and dominant, or autosomal and recessive, or it can be sex-linked and dominant, or it can be sex-linked and recessive. Now, right away, we can eliminate the dominant choices. And the reason why we can eliminate the dominant choices is, notice grandma and grandpa here do not have the trait. So if the trait is autosomal and dominant, they must be homozygous recessive. Well, then where did their kids get that trait from? That's not possible, okay? So we can rule out, and for the same reason, we can rule out X-linked dominant as well. For the X-linked dominant to show up, it would have had to have been present in the parents. So we have 2 choices that are left: autosomal and recessive, and sex-linked and recessive. So I don't know of any better way of doing it other than kind of trial and error, although you can sometimes look for one part of the Punnett or the pedigree that would indicate that it couldn't be. So I'm going to assume that it's autosomal and recessive. So if it's autosomal and recessive, there are 3 possible genotypes, and only this one has the trait. So if the trait is autosomal and recessive, I know the genotypes of these 4 individuals. Let's see if we can figure out the genotypes of the others. So grandma and grandpa, 1, 1, and 1, 2, do not have the trait, so they must have a big A. But since their kids have the trait, they must both be carriers for it. Okay? Now, some of you might be saying, if grandma and grandpa are both carriers, big A, little a, then you would expect 1/4 of their offspring to have the trait, be little a, little a. But yet 2 out of 2 of their kids ended up with that trait. Is that possible? And the answer is sure, right? If I roll a die, I expect to get a 5 1 time out of 6. But I could get it. And if I roll 2 dice, could I get double 5s? Sure. It's not super duper likely, but it happens. So remember, when we do those Punnett squares, what we're looking at is probabilities of offspring. Okay? We don't have to have 4 offspring, and we don't have to have 1/4 of them be little a, little a. So far, this is consistent. Now, notice 2-1 is not the kid of 1, 1, and 1, 2. And I know that because there's no vertical line coming down to him. So he's basically 2-2's partner. So I don't know anything about his family other than he doesn't have the trait. So big A and something. And I can put in a big A and something for these as well. Now, notice that 2-1's kid has the trait. Well, one of those alleles would have come from mom, but the other one must have come from dad. So he must have been a carrier. And he certainly could have been. Similarly, 3-1 must have inherited a little a from the mom. So far, everything is consistent with this mode of inheritance. All right, over to 2-4. She is not a member of this family by blood. She's married into it, perhaps. So I don't know anything about her other than she doesn't have the trait. But notice her son does, so she must be a carrier. So what about your friend here? All I know is that she doesn't have the trait. So I do think that we can figure out she would be big A, little a because her dad had the trait. Okay, so now let's try sex-linked and recessive. If it's sex-linked and recessive, then the only way for a female to get the trait would be to be X little a, X little a. A male would only have the trait if he had X little a and Y. Okay, so I think I can do this since it's a different color and we can see. So grandma and grandpa, she has 2 X's, he has an X and a Y. He does not have the trait, so he must have a big A. Grandma does not have the trait, so she must have a big A, and I'm not sure about this second allele. Now, when I get down to their daughter, 2-2, she is female, so she has 2 X's, and she has the trait. So she must have 2 little a's. One of them could have come from her mother, so that's possible, but where did the other one come from? Dad does not have a big A, a little a to donate. All he has is a big A. So this mode of inheritance I know is wrong. So I've already eliminated the dominant ones. I've eliminated the sex-linked recessive one. So the only mode of inheritance that is consistent with this would be autosomal and recessive. Okay. So if your friend here says, hey, should I have kids? You need to look to see, all right, I know that you're heterozygous. You got a 50-50 chance of passing on that recessive allele. And whether her kids would have it would also depend upon who she chooses as a partner to have kids with. If her partner is big A, then none of her kids would have it. Half of them would be carriers, okay? So basically, this is what genetic counselors do when people have a history of some genetic condition. Genetic counselors would go through, figure out how the trait is inherited, and then counsel those individuals as to whether it's a wise option to have kids or not. Okay, so for a pedigree, you just need to give it a try and see. All right, are there any questions about any of the genetics problems? Are you starting to get the hang of them? So I'm not hearing too much. Let me give you some advice. I've given you the genetics practice problems, the genetics extra homework, and those are the types of problems that you should be able to do for the next exam. So as with all of the exams, the time that you're allotted is limited. So if it takes you 5 to 10 minutes to do one genetics problem, you're going to run out of time. So that extra genetics homework was extra practice so that you can become a little bit more proficient in them, okay? So the time has been adjusted so that you will have time to do your Punnett Squares, but if it's taking you forever to do them, then you need to continue to practice until you can do them in a reasonable amount of time. So if it's taking you 5 to 10 minutes for a question, you probably will be short on time on the exam. So that's why we've given you extra practice. The other reason I've given you extra practice is, as I said before, genetics is a topic that I know better than most. And I've been teaching genetics for, well, 30 years. And either you get it or you don't. And it's like going over a hump. If you don't get to the top of the hump, you just kind of slide back and everything is kind of muddy and you're not sure. But once you get to the top, then it's easy sailing down to the bottom. So I strongly recommend that you continue to practice with those genetics problems until you get to that top of the hill where it just, oh yeah, now I'm getting it. So that extra little bit of practice definitely should help improve your ability to do these genetics problems. If you follow the advice that I've given you with the separate Punnett Squares and you pay attention to what you're doing, they really aren't that bad. And the more you do them, the faster you get at them. So by all means, take that extra genetics homework seriously and really use it as a practice for the test. Yeah, so as you can see from the chat that Professor Hoffman put in, there are fewer questions and given a little bit more time, because it does take a little bit more time to do a pedigree than just answer a multiple choice sort of question that's, reading the question and figuring out the answer. But again, if it's taking you a lot of time to do each question, you will run out of time. So you do need to do your best at practicing. Yeah, so absolutely you can do a Punnett Square for blood type. So let me show you. So you've got someone who is AB. So for blood typing, there is a fancy notation for the alleles, and the fancy notation is, this is the A allele, capital I with a capital A superscript, capital I with a capital B superscript for the B allele, and a lowercase I for the O allele. I tend to be lazy, and so I just use these for my alleles, okay? So that's fine. Now, I notice you put AB positive and O positive. The positive is totally separate alleles from the A, B, and O. Totally different locus, assort independently. So we can't consider it at the same time. We need to consider it separately. So in this case, you have an AB parent. The only genotype possible for AB is AB. And then you also have an O parent. The only genotype possible for an O person is OO. So you just do your Punnett Square. If one parent is AB, they will pass on either an A or B. The other parent passes on an O. And so the two possibilities for their kids would be type A and type B. So AA and AO are both type A. So A is dominant to O, okay? So AA or AO gives you type A blood. So these would be type A blood. The bottom two would be type B blood, okay? Now, in your particular problem, you said that both parents were Rh positive. Well, the Rh positive is a dominant trait. So I don't know whether they would be big R, little r for the Rh factor, okay? And oftentimes we use the letter D for that, but I thought R would make more sense for Rh factor. So because they have the dominant trait, I don't know what their genotypes are. So I can't figure out that. So it is possible for two people that have Rh positive, right? So you have AB positive and O positive. It's possible that the kid would be positive or negative, right? If they were both carriers. If you have a negative and a negative, you will only get negative blood type, okay? And as you can see, your question about producing an O kid, the answer is 0, none of them. Yeah, thank you for typing that in, James. My penmanship with this stylus is not the greatest. Yes, exactly. So if you have an AB parent and an O parent, they will not produce any type O children. They will produce type A and type B. Any other questions? All right, so my suggestion, since I'm not hearing any more questions, is definitely look over the Mastering Biology for the genetics problems and the extra genetics homework. The extra genetics homework is worth 16 points, as Professor Hoffman pointed out, so you definitely want to do it because you don't want to lose 16 points. Plus, it's great practice. It is the closest you're going to get to a review sheet for the exam that we're going to have. So definitely take that seriously. Those are the sorts of problems that you would be expected to do on the exam. So if you're struggling with any of those, you can always email me or Professor Hoffman, and we can answer you that way. And if necessary, we can do another Zoom. But hopefully this has helped you with figuring out how to do your genetics problems.
Rick, I see on your PowerPoint you have a 2 and a 3. Were those to review? Because we didn't do those yet. I can't read what the questions are because it's too small on my screen. but we did the pedigree chart, and then you have 2 other things as well that we haven't done.
The charts on slides 2 and 3. Oh, this was just more practice for DNA stuff. Is that what you're asking about? Oop, I better go back to annotate.
Yeah, we haven't done that yet.
Yeah. So this is just a practice that I came up with for the DNA stuff, but yeah, I hadn't covered that yet. You probably should be looking at that in the lecture modules by now. I'll have to look at the schedule to see, but yes, you should be considering how to do the DNA and protein synthesis stuff from the lectures. Yeah, this Zoom was only about the Punnett squares and genetics, not the central dogma stuff.
The central dogma lab is due today, everybody, though, so you should know how to do it, because I believe, if I remember right, this is covered as part of the lab, very similar chart and tracing these things out. And that's due today. So hopefully you've done it already, as Dr. G mentioned, but if not.
Yep. All right. Anything else? Anything you need to add, James?
Nothing. Just a reminder, and because you're all here, you're probably the people I don't need to talk to. But the schedule, right? I posted it, and literally every day for the next 15 days, except for 2 of them, you have something to do, at least one thing in class, right? So just 2 more weeks. Hang in there, keep pounding away, and it'll be done before you know it. So you know, just keep working hard.
Yeah. If you've made it this far, you're on that downward slide that I talked about, where the finish line is in sight. So hang in there. And also, I didn't check yesterday, but the extra credit that I posted for the iNaturalist, those are, in my opinion, 10 pretty easy to get points if you just spend, I don't know, maybe even a half an hour. It's certainly less than an hour, and it'll be sort of a fun activity. So I would recommend that you look at that at some point, maybe over the weekend, who knows. I think that was due next Saturday, but no reason why you can't turn it in a little bit early either. Okay, get some extra credit and help you out.
The iNaturalist is due by the 18th, right? And as always,
Yeah, I
we
think so.
recommend you get it done early.
Yep.
Anything else? All right, so it looks like the recording is still recording, and it will be rendered and posted as soon as it's done. Otherwise, if there's no more questions, we'll thank you. Feel free to email us and get busy, I guess.
See you, everybody.
See ya. Bye.
You have a great day.